Thank you, Elliot. We are right on the hour, so I will direct questions to the chat and move on to introduce Eneida Hatcher from NCBI Virus, who will talk about their resources for monkeypox and orthopox virus.
Hello, everyone. So hopefully you should be able to see my screen now. I'm smiling because I was in Elliot's lab and it was nice seeing all the faces from the lab that I used to be part of. So I will be talking about monkeypox and orthopox data and tools from NCBI Virus. And I have my email provided here. So when the slides are shared later, feel free to contact me if there's any questions or suggestions. And I won't go over the agenda too quickly, but so what I wanted to start out with was the NCBI Refseqs. These are reference sequences for a species. We try to work with the ICTV to develop like which sequences should be used as Refseqs. These are pulled from the GenBank database. So INSDC is a collaboration of international groups. When one sequence is submitted to an INSDC database, they're shared by all of them overnight. So they all have the same information. The Refseq sequences are always based on publicly available sequences from INSDC. And when possible, we try to work in collaboration with community experts to update the sequences. So we do things like look at publications to try to find more information on the source, so the host or the country, if those are not provided already on the record. We also try to update things like the annotation on them. So you'll notice for the monkeypox virus species, I listed the accessions for the clade 1 and clade 2 Refseqs that we have now. The clade 1, obviously, the formerly called Congo Basin or Central African, and then the newer one that we have is based off of clade 2, or formerly called the West African clade. Both of these Refseqs now have OPG gene names. So these were proposed by Sankovic et al. In a publication, and I have the links at the end, the references to the publication and all. This will help with finding homologs across different species or within a species so that everybody knows exactly what open reading frame they're talking about when we're speaking about a certain protein. I also updated the CDS or protein names, and these are based off of the BV-BRC SOGs. So they have more of a descriptive protein name. And we also provided the vaccinia virus, Copenhagen, and WR homologs to try to match up different vocabularies for the protein names that have been used historically. I mentioned the CDS annotation, and that the annotation is somewhat characteristic of GenBank records, the names that are used for the annotation. So here's some examples of what that looks like. So a gene is just called a gene in the annotation. So for example, it'll have the slash and then the word gene, and then the name of it is provided directly afterwards. So for example, OPG019. A lot of people tend to use the locus tag, which is not as reliable as a gene name. If there's a gene name provided, I would stick with that. The CDS or protein open reading frames have a product that provides the name, and in this case, the more descriptive name. And then we also included a note. This is also from the SOG database. So we have the taxonomic breadth where that protein is found, some basic information on it, and this is where we have the vaccinia virus homolog names. If we find, or if we believe that what used to be an open reading frame in an ancestor of the virus is possibly no longer expressed, so it's a fragmented gene or it's truncated, then I have annotated it as a MISC feature in the GenBank record. So, and then the name of the homolog is provided as a note. And then we also have the repeat regions for the inverted terminal repeats annotated on both of the Refseqs. So I have an image here at the bottom that shows a little bit of how these MISC features work, the way that I did them. The first two sequences at the top are monkeypox virus. The bottom two are cowpox viruses. So you can see that there's this big deletion in the monkeypox viruses compared to the cowpoxes. So it leaves only pieces of the open reading frames. So I annotated the entire piece that's left, which does often include stop codons. And I gave it the name so that you can tell when something is partial compared to what the normal protein looks like. All right. If you're interested in downloading sequence data, and this is again from the GenBank database, so from all of the INSDC partners, we have a monkeypox virus data hub. The way that our data hubs work is it's our normal NCBI virus table-based interface that you see here on the screen. When you select a certain species, then sometimes there will be a data hub header that pops up, and that has related interfaces, quick links to other resources that you might be interested in. But all of the other features are the same regardless of which species or taxon group you're interested in looking at. So the fastest way to get here is to just do a web browser search, a Google search for NCBI virus, and then you can see the top filter. Oh, my arrow's not working. The top filter is the species or the taxon group that you want to look for. So you just enter monkeypox virus there. There's various filters that you can apply. I think some of the most appropriate ones for monkeypox virus are you can search by sequence completeness or for a minimum or maximum length of sequence. You can search by protein names, and then you can download either nucleotide sequences that contain that protein or just the protein open reading frame itself. And soon we will be adding the clade and lineage classifications using the Nextclade tool. So you'll be seeing that as a filter as well as a column that's in the data. Once you've selected the data set that you're interested in, then you can quickly build a alignment or a phylogenetic tree. This is limited to a certain, a maximum number of sequences, so you can't do all of the monkeypox viruses at one time, but it's a quick way of taking a look at the sequences that you have and making sure that you have what you expect. Once you've got the right data set, then you can download the sequences either as a set of FASTA sequences, as a metadata table that contains all of the metadata that's associated with the sequences. So the source, the country it's from, the length of the sequence, all of that. You can get also an accession list if you're taking it to a different resource. And then something that we're currently working on is a dashboard visualization. So I like to call these visual filters because they still do work as filters for the data set. So these help you to explore the data. You can see clearly where sequences are coming from around the world. You can see how often they're coming in. So the collection date or the release date, those are the two histograms that are on the side. It helps you, I think, identify some of the assumptions that are fair to make about the data when you're doing bioinformatics analyses. We're hoping to add other visual filters in the future. So if you find anything that you think would be interesting, we'd love to hear about it. And there is a feedback button in the bottom corner of all of our resources, so you can provide feedback or questions, anything like that. Okay, we have other NCBI virus resources for monkeypox. Datasets is a newer tool. You can download entire packages or files of data. So for example, all of the genome sequences, protein sequences, metadata, annotation, all at one time. The really nice thing about it is that they have a command line tool so that you can plug this into a script that you're already running instead of having to visit a website, but they do use the normalization. So all of our metadata from NCBI virus is normalized, and they use the normalized metadata through data sets. Also, if you're more interested in short reads instead of assembled data, then you can visit the SRA short read archive as like raw sequencing data. And I provided the URL that'll take you there pre-filtered for monkeypox sequences. I wanted to talk briefly about submitting data. I know that sometimes this can be a challenge if you haven't done it before, or sometimes even if you have done it before. So for submitting to GenBank, so your assembled sequences, the submission only needs to include your contact information and contact information for any of the other authors for the sequence, the FASTA sequences, and metadata. So things like isolate name, collection date and location, the host, all of the information that people downstream are going to be able to use when they take advantage of the data that you're providing for them. Currently, we are not requiring the addition of the feature annotation. So the gene encoding regions for monkeypox sequences, and that's so that we can expedite their release. I'll say I'm not part of the GenBank submission group, but they really are trying very hard to listen to customers and remove some of the pain points when it comes to viral submissions. If you are interested, if you have data that you want to submit, you can go to the Bankit web interface or use the table to ASN command line. And I provided a link to some documentation on what those processes look like. Then we also have SRA submissions. So these are the raw sequence reads. And then there's a link to documentation of what that submission looks like. It's similar, but there's different files necessary, obviously. So I wanted to leave a bit of time for questions. Yeah.
Thank you for that, Eneida. Let's take a look at the question, the chat, and you guys can also feel free to unmute yourselves to ask questions. It's not often you get to directly interact with NCBI, so take advantage of the opportunity.
So I'm going to open the chat too, just in case.
Sure. So I have a question, Eneida. You mentioned that there's an upper limit on the multiple sequence alignment tool and the phylogenetic tree building tool, and I'm just curious what that number is, given how large the pox virus genome is.
Yeah, it's usually around 500 sequences. I will say that for the pox viruses, if you're doing an alignment, I would limit it to maybe 50. It'll get done. It just takes a while to get done.
Yeah.
Yeah.
And what
That's not unexpected.
and what algorithm or tool are you using for making the multiple sequence alignments?
It is an in-house tool that was developed a few years ago. So it's something like a muscle alignment,
Okay.
but the code for it has been changed around. So I will like the alignment and the publication, they're good for exploring the data and verifying that you have the type of diversity that you expect, or maybe that you don't have like a bunch of ends in your sequences if that's not what you're looking for, but they're not like publication quality alignments or trees.
Okay. And then it looks like we have a question from Kevin Liwit asking, for FTP submission, where can we find documentation regarding the specific wizards and standard metadata required for GenBank submission?
Okay, so FTP submission for viruses is, I think, only influenza and SARS-CoV-2. So not for monkeypox virus yet. And I will try to share the documentation in the chat when I get off the video.
Thank you.
Yeah.
And then another question from Richard Sherman. Are there plans to have Refseq sequences for both the 2A and 2B subclade?
Okay. Yeah. So one of the things that we try to do with the Refseqs is limit the number of them that we have. So it makes it easier for people to identify which Refseq they want to use for their purposes. However, if people come to us and say that they're having trouble using the Refseq in order to like assemble sequences or something like that, or if they're having trouble using the Refseq in a phylogenetic tree because it's not close enough to the sequences that they're interested in, then we're certainly open to creating another Refseq. We try to avoid it just to prevent any confusion. But yeah, if there's a need, then we're certainly open.
Okay. Oops, sorry. That sounds good. So there's a couple more very technical questions that you might want to get into in the chat.